Great. Good afternoon. My name is Maddie Bowers, and I am a graduate student in the GMU Forensic Science program. I'm honored to introduce the following speakers in the panel: use of forensic anthropology and forensic genetic genealogy in investigations. Dr. Joseph A. DeZino is a faculty member in the George Mason University Forensic Science program, where he teaches graduate level forensic science courses and serves as the GMU Forensic Science program graduate research coordinator. Prior to his arrival at GMU in 2012, Dr. DeZino served as a special agent in the FBI, where he served as a field agent for 3 years, followed by approximately 20 years of service in the FBI laboratory, where he served as the FBI laboratory director when he retired in 2008 after 22 years of service. Dr. Anthony B. Falsetti, forensic anthropologist, received his doctorate from the University of Tennessee, Knoxville, and is a board-certified forensic anthropologist and the identification unit supervisor at the Office of the Chief Medical Examiner, Washington, D.C. Dr. Falsetti is the forensic anthropologist for the National Missing and Unidentified Persons Program, providing skeletal analysis for medical examiners and law enforcement agencies nationwide. He has nearly 35 years of experience as a forensic anthropologist, focusing on skeletal analysis, bone trauma analysis, and the identification of unidentified decedents. Dr. Falsetti has been deployed to many mass fatality situations, including commercial airline mass disasters like the American Eagle and TWA 800, domestic terrorism like the Oklahoma City bombing, foreign terrorism, the World Trade Center, and natural disasters like the Thai tsunami and the Haitian earthquake. His research interests are bone trauma, bone pathology, new methods for positive identification, and postmortem interval analysis. Throughout the last decade, CeCe Moore has been considered an innovator and pioneer in the use of autosomal DNA to resolve unknown parentage and family mysteries. CeCe has been appointed a 2019 to 2021 non-resident fellow of the Du Bois Research Institute at Harvard's Hutchins Center. She is the chief genetic genealogist for Parabon Nanolabs, heading their genetic genealogy services for law enforcement unit, boasting the unparalleled record of over 200 successful identifications of violent criminals in the 2 years since the unit's launch. Her work has led to the first conviction, the first conviction by jury verdict, and the first exoneration in criminal cases where the suspect was identified through investigative genetic genealogy. Dr. Mark Wilson is a retired supervisory special agent with the FBI and is now an associate professor in the forensic science program at GMU. His area of expertise is in forensic DNA analysis, including DNA sequencing, mitochondrial DNA, probabilistic genotyping, and interpretation.
Thanks, Maddie. So we have 3 excellent speakers, and they can talk forever about what they do, so I don't want to take much of their time. Dr. Falsetti will lead us off to talk about forensic anthropology. And then we have 2 speakers, Dr. Wilson and CeCe Moore, who will talk about forensic or investigative genetic genealogy. And I think you'll really find their talks very interesting and see how the two disciplines may overlap, not only on old cases, but on current cases. So with that, I'll give it to Dr. Falsetti. Thanks.
Thank you, Joe. Thank you, everyone. I will try and keep this brief because I know CeCe Moore is who you're really here to see, but thank you. So I am at the Office of the Chief Medical Examiner here in DC. And great. Am I supposed to do something? Okay. Okay. Can I use this? Oh, excellent. And yes, I did study at the University of Tennessee. They give you this tie when you leave the University of Tennessee, orange and white. I was very lucky to have that opportunity to be at the University of Tennessee, working with Bill Bass, Dick Chance, Fred Smith, and the folks who really, put us on this path of identifying the unidentified. I did get to spend some time in Long Island, did a postdoc at SUNY Stony Brook, and I am board certified in forensic anthropology. I'm at the Office of the Chief Medical Examiner, so I've kind of tailored this more for this audience. For those of you who've not been to our office, we are in Southwest DC. We are on E Street. We are within the Consolidated Forensic Science building in Southwest. On one half of the building and on most of the floors is the Department of Forensic Sciences for the District of Columbia, and we are on floors 5 and 6. One of the few places where we have our actual autopsy morgue and everything on the 5th floor. The 4th floor's not all that happy about that, but that's where we are. Our structure is we have a chief, we have a deputy chief, and we have currently 4 medical examiners and 2 fellows. They're interviewing for a 5th as we speak. And our chief does cut on occasion. So that's Dr. Diaz is our chief. We have medical legal investigators who go to the scene. They document the scene. They will bring material back that's relevant towards the identification if it's on the individual. All other material of value goes to law enforcement. And we have ID specialists. I have a team of 5 individuals, and I am a unit of 1 as an anthropologist. We have our autopsy assistants who do most of the cutting. We do have a team of photographers, and we do have a transport team. We transport our own decedents to and from. Our transport team also runs throughout the city and collects X-rays and things for identification purposes. Our office is 24/7. We have 14 medical legal investigators, and we cut on the weekends. So it's a busy office. So it's not a small operation. Some of the autopsy suites, just to get a, we have 1 large suite with 5 stations, 3 other smaller single suite rooms, and a BSL-3 laboratory. Just to give you some sense of the caseload, I know this stops in 2020, but just imagine the line just keeps going up. In 2020, we had 7,600 deaths reported to the office. Obviously, not all of those are autopsied, but a fair number are. This past year, my first full year, we were in the 4,000s in terms of cases. Right now, we're at, when I left the office this morning, it was case number 2374. So those are the cases that are actually in the office. So it's a busy office for certain. By policy, everyone who comes into the office has to be identified scientifically. If your family member is with you at the hospital and you die and they say that's you, that's who you are, that case is unlikely to come in anyway. If in any other situation you are otherwise unidentified or even tentatively identified, you must be identified scientifically. And that's what my unit does. The majority of the IDs, as you can imagine, are by fingerprints. Given the population of the District of Columbia and the subpopulation of people who wind up in our office, many of them have been fingerprinted before. So that's often the majority of how we ID people. But as you'll see, decomposition is something we deal with pretty frequently. X-ray comparisons, again, that's where the role of the anthropologist comes in, without going into great detail, but there is a cooperative of all the hospitals within the district, and they contribute online to medical records as well as X-rays. So we have access through permission of vital records. We can tap into that and pull electronically X-rays and actually do a lot of X-ray comparisons. That's one of the things I spend the majority of my time doing. Dental is fairly infrequent. We've only had a couple where we've had dental comparisons, but we do have access to an odontologist. And we do DNA. Again, fairly rare, but it is one of the ways in which we can identify unidentified decedents, and includes genetic genealogy. And that's kind of where I'm going to talk about today. Forensic anthropology. We study bones. We apply that knowledge to medical legal investigations. Pretty straightforward. What do we do? Most of you who are medical examiners are fairly familiar with what we do, but we look at unidentified decedents, primarily skeletal materials where this is derived from, and we try and do three main things. Obviously, the biological profile, we want to develop that for purposes of identification. We want to determine the postmortem interval where that's important, hence the body farm, not too far from here. And we want to gather information about the death event. And this is what will eventually tie into the genetic genealogy. What can we learn about that individual? What's revealed about that individual through their bones? What can we tell about them that would add to that profile besides how old they are? What is their biological sex? What else can we do? We look at evidence of trauma, either remote trauma that has occurred in the past or acute that occurred at around the time of death, and then life history events. These are the kinds of things that we try and build the profile as best we can from an unidentified decedent. Again, it's the basics. Age, how old are they? Many indicators on the human skeleton prior to about age 25, in which we could look at epiphyseal closure of the ends of our long bones, things of that nature. After that, in our 30s, we're looking at changes that occur in the pubic symphysis, the ends of our ribs, and there are standards in which anthropologists apply to that to get age ranges. Sex, again, is based on skeletal morphology, ancestry, also based on skeletal morphology, in particular the skull, and even more specifically the face. How tall someone was, it's fairly straightforward. And then acute versus remote injuries, again, building that profile out a bit. And then postmortem interval estimates. The kinds of cases that I work on are just anything you can imagine that occurs in anywhere, but in the district in particular. We do get a lot of decomposed bodies. There's a lot of squatters in DC, if you weren't aware of this. A lot of properties that are otherwise unoccupied are actually occupied by persons that don't want you to know they're there. We do get burned bodies. A lot of traffic fatalities that come in subsequent crash fire. Those are remains that we deal with. Buried, not very often. Although there is a lot of green space in DC, there's not a lot of place to actually bury a body, although I have recovered two just in the past 18 months that were buried in DC. So anytime you're in a national park, kind of look on the ground, see if you see any evidence of a grave, because it could be there. Partial skeletons, certainly, and just, you know, the whole list. We do a special kind of survey in our office. It's a skeletal survey. Any individual who's 2 years and younger gets a special radiographic examination, as well as full posterior cut down, looking for acute versus remote injuries. And that falls to the anthropologist. So my experience with genetic genealogy, to use a case, and just kind of walk you through this one. On the 4th of October in 2020, a decedent was brought into GW Hospital, was there for 3 days, died. This person was not decomposed. They had fingerprints, plenty of dental characteristics. No ID, no earthly idea who he was, no surgical interventions, none of the sort of biological clues that we might look for. You know, no internal fixation devices where we could go to this database we have and sort through that. He had no ID on his person, and like it says, some ring camera captured him going into this location, but no information on that. And so, you know, very soon after, homicide is looking for this individual. About a year later, get the DNA back. from the FBI, no hit. Another year, we sent a blood card to the DNA project, and they developed a profile. About a year later, so this is just a decedent. Otherwise, you would think would be fairly easy to identify. It's not a tricky skeletal case. It's nothing like that. It's just an individual found down, taken to GW hospital. The DNA project makes a distant connection via ancestry.com. And so throughout this whole process, the several-year process, I went back in and we were able to look at this individual more closely, did some excisions, found serial rib fractures that had healed. So he had remote rib fractures. He also had acute rib fractures. So this was someone who'd been around on the streets, so had run into instances of interpersonal violence. And so that goes into the building of the profile. He's been in NamUs. He has dental charts. He has everything. And until the DNA project took over, we had no idea who he was. 84% African American. You can see where the distribution is, and again, this may change as more databases come up. They've got it to the point where they're looking at certain surnames, and we were looking at Washington, D.C., Maryland, and potentially South Carolina. They got it narrowed down to the counties. These remote rib fractures will hopefully help. He does have dental characteristics. He has dental records. So we were able to, all of those go into his profile, along with his DNA. We had an artistic recreation. That's just basically his autopsy photo that's been colorized. So he was otherwise visually identifiable, except he wasn't. No one knew who he was. So this is where he is now. This just went out last week. So we're hopeful as these databases become more populated. I'm sure Mark and CeCe will tell you more about the nature of those databases. But from an anthropological point of view, someone responsible for identifying the unidentified, at least in DC, we want to look and leverage our partners out there who are doing this kind of work. It is an interdisciplinary approach. In this instance, we knew how long he'd been dead, so the postmortem interval wasn't really a question. His remote injuries were well healed, but still evidence of scarring, so we knew they had occurred. The acute injuries must have occurred right around the time of death. He's considered a homicide by law enforcement. And so when we combine, sort of the anthropological methods going in, ascertaining the age, the age assessment was done at the hospital by the doctors, which they just looked at him and said, well, he's such and such. Well, maybe not. So literally did a little more digging. We're able to not only look at his rib ends, but looked at the pubic symptom and narrowed that age range down. They had overestimated his age. I think it was because of his beard and there was a lot of gray in it. So they just automatically upped that age. And we were able to bring it down a little more reasonably, at least based on a skeletal structure. And we were able to combine that with, this offer for genetic genealogy. And, we can combine both the biological and the sociocultural. The District of Columbia is, even though there are tourists there daily, it's still a very small community of people who live in DC and in the greater Maryland, Northern Virginia area. So as that population has access to these databases, and you have to look to see if folks are willing to put their data in these databases. That we'll be able to narrow down who this man is. And we should all review the challenging cases as new methods and resources become available. As data comes out of the body farm here, we can revise these PMIs. Work that Mark's doing, he's going to tell you about, and genetic genealogy. These are new methods and new resources that are out there and they're available. The DNA Doe project actually came to us because they wanted to do one of our cases. So I'm going to leave you pretty much with that. Also put in a brief plug for NamUs. I'm sure you all know what it is. They do have Anthropology, and that's actually me. Those cases will come to our office free of charge, and we'll work them up for you. But they also do some, they have access to databases where they can, at least on unclaimed bodies, they can do. We've had some unclaimed bodies be claimed ultimately by finding family using the NamUs system. So I'm going to leave plenty of time for Mark, and I guess we're going to do questions afterwards. Okay. All right. Well, thank you all very much.
Okay. Thank you for the invitation. It's an honor to be here. I want to go through a lot of information here. It's going to be kind of fast, but I know you're waiting to hear CeCe. I'm going to bounce back and forth between policy issues and a little bit of science. I'm going to tell you a little bit about the DNA, how the samples start, what we look for to get the whole process started. So, so here we go. That's who I am. I'm at GMU, and okay. So here's part of what I'd like to cover. In forensics, of course, it's very important to have good quality assurance, quality control. This new emerging technique of investigative genetic genealogy is considered to be background research or investigative leads. So it doesn't usually see the inside of a courtroom. It's considered background. And so the question is naturally, what are the appropriate standards to apply to this, like personnel certification, laboratory accreditation? That's all now being worked out under this general idea that it's background information, it's research information. However, there are unique issues associated with this because it relies, as you'll see, on the use of private databases. GEDmatch and Family Tree DNA are the two that are used by law enforcement, and they're owned by private companies. There's no government oversight of them. So policy-wise, they could decide to change their policy because it's a private company and no one could say anything about it. They could go out of business. If they both go out of business, decide they don't want to be in the forensic arena anymore, then what? What do we do? So this is another consideration to think about in terms of the long-term viability of genetic genealogy. I want to also talk about some funding resources available to you should you be considering using investigative genetic genealogy in your casework. I'll tell you about where to find money to do that. Okay, so what's different about this technology is there's just a lot of information. So for example, over here, the genotype listed here, you can see that there's just two letters. It's a simple code. It's a SNP genotype, it's called, but there are 850,000 rows of data. So compared to the existing DNA kits that are used, which have typically 20 to 27 markers, this has hundreds of thousands of markers. So it's very information rich, meaning that you can do a lot more with it. You can predict a person's physical phenotype, what they look like. You can do deep background ancestry and tell admixture from a person's ancestry. All this factors into the final genetic genealogy report and conclusion based on all this information. And the reason that we have a lot of this information is because of the depth of all the available SNPs, in this case, to analyze. So you can see the position, that's just the position in the genome. These are all on chromosome 1, different numbers from different genome builds, and then the genotype. So keep that in mind as we go through this. There's a lot of information, and it's that depth of information that is really important. So what happens to the information is it's uploaded, it's compared pairwise to samples in the database. If there's a match, they share what's called a centimorgan. It's just a measure of relatedness. And this is typically what's found with first cousins, about 300 centimorgans. But this can then be taken and used to build back family trees. And I'll show you how that's done. So the first case was, of course, the Golden State Killer case, Joseph DeAngelo, back in 2018. So the field has been at this a little over 5 years. And in that time, hundreds of cases have been solved using genetic genealogy. So it's definitely here to stay, but there are issues as I'll go over and I've already mentioned. So this was the first case. And then shortly after that, the floodgates opened. A lot of companies got involved, and there are many identifications that were made. That led the Department of Justice to release some interim guidelines back in September of 2019 to kind of shepherd this newly emerging technology. So some of the things that they said is it's for research. They use the word research and generate investigative leads for unsolved violent crimes. They list homicides, rapes, and aggravated homicide, aggravated assaults. Another thing that they mentioned is that existing DNA data should be used, specifically STR profiles. STR profiles are the main type of DNA information that crime labs use. And there's typically 20 to 27 of these markers. And so what they said is, yeah, we'll agree that this is a very powerful technology. However, we want you to compare ultimately back to the crime scene DNA, which had the STR profile on hand. So one of the requirements is to have an STR profile in hand to compare back to. That's very important because that then underscores the fact that this research is in the background. It's not the DNA type that's relied in ultimately to make the match. It's the STR. It's supplementary or supportive of the STR type. And also it has to be previously uploaded into CODIS. CODIS is just the criminal database that contains 20 million or so profiles. And so the idea was, let's upload to CODIS. If there's no match in CODIS, then the next logical step is to go to genetic genealogy. There are privacy issues, and typically those issues deal with matching people that are in a family or might be in a family, which are called target testers. So to approach these people and get their consent to participate, the DOJ said, let's make sure that that's done. Let's make sure that everything is above board and everyone has agreed to be typed if they're going to be used in this fashion. They also stated that they needed approval from a prosecutor before covertly collecting any reference samples. So you know that discarded DNA, discarded items that are thrown out are free game to be collected and typed, but they stipulated that they would like approval of a prosecutor before that is undertaken. So target testing and the targeted kinship testing is very important in this area because it can be relatively fast. It uses a technology called microarrays. You see a microarray over here on the side. And what it can do is it can tell fairly quickly whether or not a person is part of a given lineage or not, where they fit into a purported tree with some probability. And I show this because this is just one of many technologies that can be used in this field. So what happens with a kinship tester is once the results come back, then you can eliminate a lot of the purported tree and focus in on the matches that are shown up from the kinship tester in the pedigree. So here's a typical result from kinship testing. You see in this instance, there's a very high probability of a second degree relationship. This is not possible with typical DNA STR typing. You can get siblings and parent child and of course identity, but beyond that, STRs are very limited. However, with SNPs and this technology, you can get very high confidence of particular relationships. Also, there were some provisions about removing profiles if investigation turns out to be lead to down a different path. Then what the DOJ stated was there has to be some kind of mechanism to remove the profiles from the databases. This is somewhat controversial because it's not specified exactly which databases and how that's to be documented. And then also in the genealogist notes, there's also some entries that are present there. So this is one of those things that has to be worked out. So here are some of the known SNP profile databases. You're aware, of course, of Ancestry, MyHeritage, Family Tree, and GEDmatch. These are the two that are used for law enforcement presently. There's Living DNA, 23andMe, and others. GEDmatch is simply a database that will perform matching and statistics on matching, and it has a lot of powerful tools that are used. Family Tree also is a larger database than GEDmatch, but these two collectively are used for law enforcement presently. GEDmatch has about 1.5 million profiles presently, and the policies on these two databases are a little bit different. With Family Tree DNA, the default is for your profile to be opted in. You can choose not to participate in law enforcement uploads and comparisons, but the default for Family Tree is to opt in. It's a larger database, over 2 million profiles. GEDmatch a little smaller, but for matching and for being opted in, you have to deliberately opt in with GEDmatch. You have to tell them and make a statement that you agree to be opted in. And this is a little bit dated. There's closer to 400,000 now profiles that are opted into GEDmatch. GEDmatch has a policy, if it's an unidentified human remains case, then the entire database of 1.5 million profiles can be used. One thing to keep in mind is the match lists that come off the database searches. So this could be from GEDmatch or Family Tree. They don't list any personal information and they don't list any SNP genotypes. You can't tell anything about the DNA itself, just the matches. So that's an important consideration. You're not dealing with any genetic information. You don't know the ACs, Gs, and Ts. Those just aren't present to you. All you have is the match list and the distance of those matches. So the SNP data is not provided. GEDmatch has a number of very important and powerful tools. One of them is this clustering tool. So here is this 2x2 matrix of matches, and what you can do is ask the program to cluster, to auto cluster. And the more profiles in the database that match each other will cluster in this way. So these are called genetic networks. Some are larger than others. And the trick with genetic genealogy is to then consolidate these clusters into one large genetic network. So these in blue here on the bottom have DNA in common with each other. And so by stepping through these clusters and building trees, a talented genetic genealogist can take all these networks and make it consolidated into one. So large and small clusters depend on the number of matches in the GEDmatch database. So here is a fictitious chart, and I show you this because this is it's a good way to visualize the relationships here. We have the ancestral couples on top, and then as we go down closer to the present down here we have the male suspect down here, and there are a number of possible locations for this male suspect within this large chart. So what the GGs can do is they can take one of these living persons and consider the option of approaching that person as a potential kinship tester. And if this person agrees and they sign a waiver that they agree to participate in this, then their DNA is collected, it's uploaded into the databases, and then comparisons are made. That refines this uncertainty. What we have here is a lot of uncertainty, but with the potential kinship tester now tested, that can be compared back, and then the degree of the overall degree of uncertainty is diminished and the relationships are solidified. So again, kinship testing is very important. And with cases that get close to resolution, what you'll see typically is more and more kinship testing has been done. So a technique that can rapidly genotype a kinship tester is important to keep the cases really flowing quickly. Okay, switching gears a little bit now to laboratory accreditation. So what is the actual DNA? What do we start with, and what are the organizations that are out there that accredit forensic laboratories and clinical laboratories? Because the microarray technology that I showed you is a clinically based technology. It's used a lot in clinical labs for medical diagnosis. They have their own sets of accrediting bodies. College of American Pathologists is one. CLIA is another, but within the forensic world, we have our own, right? We have 17025, that's under ANAB, and that's under the broad umbrella of ISO. So one of the questions to work through as we progress is the appropriate level of certification and accreditation for the practitioners and for the laboratories. Fortunately, though, the FBI laboratory many years ago went through and identified all the elements of quality control that apply to DNA. And so here they are. So they've laid out the elements of these and described what they mean by each of them. And so we have a good framework to start from. But now questions arise, obviously, because there's a lot of things here that might only apply to forensic typing in a forensic laboratory, full-time forensic laboratory. But if you focus just on one issue on personnel training, so if we look at personnel, who are the right people to be certified? The lab analysts that work in the lab, well, they typically are certified anyway. What about the bioinformaticists that crunch the data and come up with the SNP profiles? Should they be certified, tested? If so, how? Who does it? What's the body that's going to oversee that? Which standards should be used? For genetic genealogists, what is the appropriate level, if any, of certification? And so these are open questions that are being discussed and developed and published now. Do we need new standards to support this technology? Keeping in mind that, again, this is background information, it's research, and so it has a different level of scrutiny than, say, a typical STR case in a laboratory. Right, it's not going to, generally it's not going to go to court. However, ANAB is now expanding its scope of under ISO 17025 to include forensic, SNPs, and investigative genetic genealogy laboratory processing. This is very new. There's only a few labs that have applied and some that have received this certification from ANAB. Okay, so how about the samples themselves? What can we work on in this realm? Well, pretty much everything. Bones for human remains, hair has come a long way. Hairs are possible. The typical forensic samples: semen, blood, buccal and saliva. Any type of tissue where you can extract DNA is fair game. Bone, as I said, hair, and then touch. So touch DNA is important in a lot of different crimes, mostly violent crimes, baseball caps and steering wheels, etc. But each one of these, although considered to be challenging, has relied on or benefited from some very important improvements in the first steps in the laboratory: DNA extraction and what's called library prep, making a library from which we can then generate data. But the point here is that pretty much anything can be analyzed. Anything that has DNA in it is able to be analyzed. So and I'll talk a little bit about advances in hair analysis, where now it's possible to obtain whole genome information, the entire human genome from a hair shaft, which a few years ago was considered just inconceivable. And it relies on a new way to extract DNA and prepare a library. About 20 years ago now, the ancient DNA community came up with a new method of extracting DNA from samples based on silica spin columns and the affinity of silica to DNA. That's come a long way. And then now, excuse me, now with this technology, we're getting better and better at getting viable DNA out of very challenging samples. So if you can get typable DNA out of ancient specimens, obviously that's a benefit in forensics as well. And the state of the art is shown down here on the lower right, using what's called chaotropic salts and different buffers. You can bind DNA to the silica column and then elute it just by changing the buffer. So you can wash off the DNA and then elute it. Very, very powerful technique that is used now fairly routinely in crime labs. So here's a little bit more of the chemistry. Just by changing the ionic strength of the buffer, you can drive the DNA onto the magnetic bead, in this case, that's coated with silica. And so this is the general silica-based DNA binding technology. Library prep. The library is what you make first from the extract to actually get the DNA type from. And now it's possible from improvements in library prep technology to get whole genome sequence out of challenging bones and hair shaft. And again, this is relatively new. So because of the major advances technologically within this field, those translate into all aspects of typing, including genetic genealogy. So we're relying on improvements in extraction and library prep to drive the science of what we do in genetic genealogy as well. So when we're looking at samples for potential samples for investigative genetic genealogy, what we want to know is primarily how much is there? And then also very important is the quality. Is the DNA degraded or not? Is it pure or is it mixed? Is it a single source sample or maybe from 2 or more individuals? We want to know how it's extracted, you know, how long it's been sitting in the refrigerator. Was it extracted in water or buffer, et cetera? And the issue with mixtures is interesting because we can actually resolve, deconvolve, and separate out mixed samples. And some companies are able to do this routinely with the identification of particular contributors to mixtures. So what happens is you can take the mixture apart, you can subtract, say, the victim from the mixture, and what's left over is the suspect, and then that suspect profile gets uploaded and run. So mixtures can be analyzed. DNA degradation is a huge issue across all aspects of forensic DNA. Over time, DNA will slowly just fall apart. It's mostly environmental insults, not so much time. In the right environment, a DNA sample can persist for thousands of years, but depending on its local environment, its microenvironment, the pH, the amount of sunlight, water, microbes, et cetera, it'll start to fall apart. And after a certain amount of time with these environmental insults, what you have is what's shown on the right, highly degraded DNA. So what do you do with degraded DNA? Well, there's new techniques that I'll talk about in a second that can help us analyze highly degraded DNA. There are, with degraded DNA, there's 2 different technologies, but the field is moving more towards this technology on the right-hand side, DNA sequencing, because the sequencing technology can capture very small pieces of DNA and generate a sequence, and that's shown on the right. It's not very long, just a few bases long, but still with enough samples in a library, you can put together a lot of these little strings and recreate larger segments. With microarrays, it's not as easy. Microarrays will handle degraded DNA, but there's a lot of missing data from microarrays. So degradation affects both technologies. However, sequencing and advancements in DNA sequencing technology are able to overcome most of the limitations with degraded DNA now. So it's only getting better. Here's just a picture of what was found with ancient DNA. It's a scanning EM showing the cross-linking, cross-bridges, the degraded structure of DNA from these forensic and ancient samples. We have a way to estimate the amount of degradation. It's a quick spectroscopic amplification technique, and we calculate a degradation index just by a ratio of the amount of DNA that's comes from a small target over a large target. And this is employed in crime labs throughout the world. So it's easy to calculate a DI, and from the DI, then we can decide what's the appropriate technology. Is it sequencing or is it microarrays? We ask a lot of questions. There's a lot of things to figure out about the sample. It's important to spend a lot of time analyzing the sample and looking over, assessing what's been done with it, how long has it been sitting around, and what buffer, has it been frozen and thawed a lot, et cetera. So there's a lot of information. And speaking of information, there's a lot of information from the human genome, right? So we're talking about over 3 billion base pairs of information and about 1.5 million SNPs total. So there's a lot of SNPs in the human genome that we can look for and analyze. So it turns out that in the human genome, there's a SNP every approximately 1,000 base pairs. So this is a string of letters that's exactly 1,000 base pairs, but there's only one SNP I show in the center of an A. This is typically the density of SNPs in the genome. You might think, well, 1 in 1,000, that's not much, but the human genome's huge. So because of that, there's a lot of SNPs that are present. But the trick is to get them out, right? You can sequence everything, but most of that's not going to be important to you. What is going to be important is the characteristics of the SNP position itself. And again, we're only looking at a subset, so you can tell the difference in numbering here. We're just focused on previously identified SNPs that human geneticists have been looking at for decades. And so there's a very large database of where these SNPs are in the human genome. And that technology has led to our ability to do genealogy. So the cutoff typically for the amount of DNA and which technology it goes to, if it's less than a nanogram, generally it's sequenced. If it's more than a nanogram, then we use the microarray. Microarrays tend to be faster, and they also tend to be less expensive. I already talked about kinship testing, but I wanted to circle back on this to again point out the fact that the microarray is a very good tool to be used in kinship testing because it's relatively inexpensive and relatively quick. There's two different types. There's many different types of arrays, but in forensics, we use the 850K high density chip. And then there, also for kinship, we use a lower density array called the GSA array. Here's a different view of the technology and the instrumentation. So on the left, we have an array scanner. The bead chip is the array that's loaded with DNA, and then it's scanned. On the sequencing side, we have a flow cell, and that comes out with a lot of information. And again, although sequencing technology is changing quite a bit, sequencing is still considered more or is more expensive than arrays. The library is very important, and I listed different colors of libraries because there's newly emerging ways to make libraries. There's a very powerful way to make a library now from very short segments of DNA, and that allows investigators to capture really tiny pieces, little snippets of DNA from a hair shaft. And if you get enough of those, you can put together the entire genome of a person from hair shaft. This was, again, inconceivable a few years ago, but now it's fairly routine. It's expensive and specialized, but it's very powerful because hair evidence is found in a lot of crimes, and for many years, not much could be done about it. But now, with this advances in this technology, with all these different library techniques, we can now feed the sequencers and generate a tremendous amount of information. There's also newly emerging technologies. For example, the nanopore sequencer. This is just a cartoon of a piece of DNA that runs through a pore. And as it runs through this pore, there's changes in the currents, and those are measured. Changes correspond to a sequence. So what comes out is a sequence. And so this is limited utility so far in forensics, but for resolving mixtures and for generating a lot of information from a small sample, this is a technology that's ready to be implemented not only in crime labs, but also in SNP genotyping as well. Okay, funding. There are many different organizations, both in the public and the private spheres. In terms of the public funding, NIJ and the Bureau of Justice Assistance, from part of the DOJ, has grants available, and they're on a yearly basis. You apply for them, and then they will support your casework. There's also SACI under BJA. There's the Sexual Assault Kit Initiative, again under BJA, which provides money specifically in this case for sexual assault kits. But of course, many of these cases are unsolved, and GG can help. BJA has these strategic focus areas. Number one, you can see a reduction in violent crime. So they are primed and ready to supply money for laboratories and agencies that are ready to use investigative genetic genealogy, and they provided many grants so far. The Edward Byrne Memorial Justice Assistant Grant, also called JAG, the JAG program, is a really good source for funding. They have a very nice website and application materials present there, and they do support funding for GG. There's the website for SACI. There's a competitive grant program. States and localities can compete for these funds, but it's fairly well funded. And you can see how from the local, this is just from North Carolina, you have some cities and the State Department of Justice. So different levels of state and local organizations can receive state and local funding under SACI to address their backlog cases. And of course, they do support GG. State funding available from many different states and localities. This is just a small example of some of the states that have put out RFPs for genetic genealogy and SNP genotyping. And so this is growing quickly as well. Utah, Wisconsin, etc. There's also private funding sources. Season of Justice is a good way to apply. These are funded by private donors, and it's a nonprofit. The Season of Justice website there, you can find out a lot more. It's a nonprofit, and it actively supports cold cases, all cold cases, especially those that can be addressed with IgG. Here's a new one down here, Media LLC, EUW Media. There's a ID there, a website, sorry, a email address if you want to follow up with this person. This is relatively new. It's a media company, but they are putting out the word that they are ready and willing to support IgG. Justice Drive is another one. It's a nonprofit, and it's here's the website for it. It uses crowdfunding and a portal to help law enforcement receive money. So citizens put money in, and then are able to get the funding from Justice Drive. Many different databases. One of the challenges for the future is to integrate all these databases. So we have the IgG databases up top for the family matching, but then there's also existing databases in the federal government, NamUs, as Tony mentioned, and CODIS, of course. So just some of the many issues. These are the things that strike me as being important, but there are many more. When you put your profile into one of these databases, GEDmatch or Family Tree, and you opt in or are automatically opted in, this is a strictly voluntary system, right? So there's no compulsion. Nobody is compelled to do anything. And we want to keep it that way. We want to keep it free and voluntary. And that's the power of it. It's really, at the core level, it's crowdsourcing in a sense, in that you're volunteering to help your fellow citizens outside of government structure so far, but you're volunteering to solve these cases by providing your DNA profile and allowing it to be used for law enforcement. We want to try to keep it that way. Private companies, though, they have their own terms of service. Those terms of service have to be abided by, or should be abided by. But they are private companies, and there's very little oversight right now of their activities. The data security it is also an issue. Maintaining the secure nature of the raw data. So, for example, if an expungement request comes through and a database is asked to remove a profile, what are the provisions in place to ensure that that's actually been done? So the raw data security is still important, and keeping the individuals in the databases private from outside identification is important too. Okay, so thank you. There's my email address. I appreciate your attention. And now you're going to hear from CeCe, who's going to dazzle you with a lot of interesting cases and tell you more about how it works. She's probably the preeminent genetic genealogist in the world, developed many of these techniques on her own. Yeah, so thank you.
Good afternoon. Are you able to hear me?
Yes.
All right, great. Well, I am going to speak pretty quickly because, as always, I have a lot to say about investigative genetic genealogy. My esteemed co-presenters really laid a great foundation. So I'm going to jump right into a case study. In 1988, in Fort Wayne, an 8-year-old little girl named April Tinsley was kidnapped off a neighborhood street near her home. She was raped, murdered, and thrown on the side of the road. Horrible crime, obviously. In 2004, the individual that perpetrated this crime wanted to draw attention to the crime and started leaving threatening notes around the Fort Wayne community. In those notes, he left some biological material that was tested, and the DNA was a match to the original crime scene. So they knew this wasn't just a copycat. This was actually the perpetrator, the same person who was responsible for this crime. So 1988 was the crime, and then this happened in 2004. When we uploaded the profile of this individual, the SNP profile, as Mark discussed, into the GEDmatch database, it generated a match list. So this was our match list. This was part of it. There are usually hundreds or thousands of matches. These were the matches that ended up being key for my research, and I'll go back to this. But I first wanted to talk about, what does a match mean? Well, when you get a match, this isn't a match like you would get in CODIS. This is not an exact full profile match. It's a partial match. It is someone who shares a portion of their DNA with this unknown suspect that we're trying to identify. They have matching blocks or segments of autosomal DNA. So these are not single marker matches. This is where you have lots of SNPs in a row that are all identical. And the only reason two people would have these matching blocks or segments are if they have a shared common ancestor somewhere in their family trees. Generally, the more autosomal DNA shared, which is measured in centimorgans, as Mark mentioned, the closer that shared ancestor is in time. So this is what a chromosome browser might look like for someone who is approximately a second cousin. So you can see all of the chromosomes here in blue, and the orange segments are where these two individuals share DNA. So those are the segments that make up the match. You add the centimorgan count of each of those segments together, and you get a total amount of shared DNA. In this case, 240.2 centimorgans. Now, this is autosomal DNA that we're using. So just as a reminder for anyone who hasn't been in biology or genetics class recently, this is the type of DNA that's inherited from all of our ancestral lines. So whether you're male or female, you get this from all of your great-grandparents and back to about fourth great-grandparents. This type of DNA changes very quickly because of recombination. Our great-grandparents pass it to our grandparents, 50% from mom, 50% from dad. Our grandparents pass it to our parents, and it's blending here at each generation. These pieces of DNA from the ancestors are getting smaller and smaller. And so when it gets down to us, we are walking around with big and little pieces of DNA from each of our great-grandparents and most likely each of their great-grandparents. So this is the basis of investigative genetic genealogy. If we can determine where these bits or pieces of DNA come from, who are the ancestors that contributed them, then we can reverse engineer someone's identity based on who their ancestors are. Now, from our parents, we get 50% of this type of DNA. From our grandparents, we get about 25%. From our great-grandparents, we get about 12.5%, and so on. So at each generation, the amount of autosomal DNA inherited from our ancestors approximately halves. And it is an approximation. These are averages. You can get a little bit more from one ancestor at a certain level and a little less from another, maybe 30% from one grandparent and 20% from the other. But this is the average. What this translates to is that we inherit some of the same DNA with our relatives. So as I said, parents are about 50%, so about 3,600 centimorgans. Our aunts and uncles or grandparents or half siblings will share about 1,700 centimorgans. First cousins, about 850 centimorgans. First cousins once removed, about 425, and so on. Now note, when you get down to third cousins, you're typically sharing less than 1% of your DNA on average. So it dissipates pretty quickly. There are wide ranges of sharing around these numbers. So these are just averages. You can share quite a bit more with a second cousin. You can share quite a bit less. Maybe instead of 3% of your DNA, you might share 6%, or you might only share 1%. So that can be a little bit confusing. You have to keep an open mind. And the other reason is because there are multiple potential relationships for each level of shared DNA. So for instance, if you share about 12.5% of your DNA with someone, or about 850 centimorgans, we can't automatically translate that to one single relationship. That could be your first cousin. It could be a niece or nephew from a half sibling. It could be your aunt or uncle if they are half siblings with your parents. It could be your great aunt or uncle or your grandniece or nephew. And so for each amount of shared DNA, there are multiple possibilities. So going back to our match list in April's case, our top match was 114.5 centimorgans. So you can see this isn't a really close match. Let's look at what it means. We can go and take this amount of DNA, this amount of centimorgans, and plug it into this awesome tool called DNA Painter, shared centimorgans. And you can put in 114.5, you see there, and it will tell us the possible relationships and the probabilities that it's one relationship over another. And so this amount of DNA is most likely, the highest probability, is going to be a 6th degree relative, which includes second cousins once removed, half second cousins, half first cousins twice removed, first cousins 3 times removed, and so on. But you can see there are many other possible relationships. There's about a 10% chance it could be a second cousin or equivalent. If it's a second cousin, that would mean that they would share great grandparents. If it's a third cousin, which is also possible, then they would share great great grandparents. So this tells me how far back I need to build this matches tree to potentially find the common ancestors with our suspect. Now, I had a lot of matches around this level. You can see I had about 4 around 100 centimorgans, 2 in the 90s, 1 in the 80s. Those are all going to be around third cousins, second cousins once removed. You can look at my next column, it starts dropping down pretty quickly now. And the furthest out match in this case that I found helpful was 49.4 centimorgans. So let's look at what that translates to. So this level of match is most likely going to be an 8th, 9th, 10th degree or more distant relative. So you can see as the amount of centimorgans gets lower, the relationships are going to be further out. So this is most likely going to be a half third cousin, a third cousin once removed, but there's also a high probability that this could be a fourth cousin once removed or more distant. So as the amount of DNA gets smaller, it gets more difficult to predict the likely relationships. Now, with this level of relationship, it can't be any closer than a second cousin once removed. You can see at the bottom, second cousins have a 0% probability. So there's many possible relationships for each amount of shared DNA. The only exception is parent-child and full siblings. Those are the only ones that we can know just from the amount of DNA alone and the inheritance pattern. So we won't know what these relationships are with these matches and the suspect until we've ID'd him. So we've got it to, as I said, keep an open mind. So our main job as an investigative genetic genealogist is to build trees. And fortunately, that's something those of us who do this love. And so we can do it all day long. Now, one of the keys is we really want to do this without contacting the matches. So what do I mean? What I mean is that it's really important that we maintain the confidentiality in these cases. And so we don't want to reach out to the matches if possible, because it introduces risk into suspect cases. And for Jane and John Doe or unidentified human remains cases, it can lead to unintentional death notifications. We could be letting an individual know, a relative know, that one of their loved ones is deceased. And we don't want to do that while we're still in the process of the investigative genetic genealogy analysis. And we also want to make sure that that goes through the official agency that is in charge of that case, rather than it coming from a genetic genealogist. So you don't want to reach out to these matches, especially closer matches. We don't want them sitting around the Thanksgiving table and mentioning they've been contacted on one of these cases and have that perpetrator get the heads up sitting at that table, for instance. And so the way we're doing this is we're using public records to try to identify who these matches are, who the DNA contributors are, and then who their parents are and grandparents, and build their family trees. We're using obituaries, newspaper articles, census records, vital records, published family trees, etc. Some of the must-have resources for investigative genetic genealogy is ancestry.com. So we are barred from using their DNA database, but they have an amazing records database that has billions, literally billions, maybe trillions now of digitized records. And those are public records, and we need those to be able to construct these family trees. As I said, we use newspaper archives, so newspapers.com and Genealogy Bank are really important. We can often find obituaries that will help us identify the members of these families and build these trees back in time. We use people search databases a lot, like White Pages Premium, or in particular, a database called Ben Verified, or BV for short. It's really important to us because often we don't have much to work with as far as who these matches are in the GEDmatch or Family Tree DNA database, but they almost always will have an email address. So this service offers a reverse email lookup. We can enter those email addresses into it, and very often it will tell us who that email address belongs to. And it will also list their relatives, their associates, people who share their address. Sometimes we can just search by their username. So if it's, say, butterfly45@gmail, if we can't find that email address owner, we might just search butterfly45, help us find that individual. We can learn a lot about individuals, their families, their associates, and that helps us structure these families. We use social media extensively in our work as well. So when people talk about genetic privacy, I often have to point out that your DNA means absolutely nothing to us if we cannot identify you and learn about your family. And so what you put out there on social media and what is gathered by these people search databases like Ben Verified is really where there is maybe something that people should look at as far as what information is available to the public. And these are all public records. We're not accessing anything that is considered private. As investigative genetic genealogists, as independent ones, third-party vendors, we don't have access to law enforcement databases either. So as Mark mentioned, genetic networks and clustering is really important in my work. So it's not just important who's sharing DNA with this subject, the person we're trying to identify, whether it's a suspect or Jane or John Doe, but who is sharing DNA with each other. Because if the matches share DNA with each other, then they should have a common ancestor in their family tree. So once we've grouped these matches and identified who's sharing DNA with each other, if we build their family trees back in time, we should identify a common ancestor or common ancestral couple. If we're able to do that, then we have one piece of the subject's family tree. Because if they're all descended from a certain ancestral couple or ancestral line, then our subject must be as well. So here's an example. Match number 1 and match number 8 share DNA. They share about 50 centimorgans in my example. And if you build their family trees back, you're looking for that common ancestral couple. And here we find them. Once we've identified them, then we know the subject is very likely a descendant of that ancestral couple. And so that gives us one piece of his family tree. So genetic networks tell us which branch of someone's family tree is relevant to our research and to our case. And so this helps us to be much more efficient. We focus in on just one branch. So when we have to build forward, trying to identify their descendants, we only have to do that from one branch of the tree rather than all sets of their great-grandparents or great-great-grandparents. So this is really important to help us focus in on where we need to be. So an example workflow in GEDmatch is first, after it's been uploaded, the file's been uploaded, and we have this match list, then we're going to determine the first genetic network by running a matrix tool. There's a matrix on GEDmatch where you can compare the matches to each other. So we take maybe the top 25 matches and we determine which are related to each other, which share DNA. Once we do that, we work to identify the top match. We'll often use reverse email lookup on Ben Verified, as I mentioned. Once we get that person's identity, we'll look in these people search databases for older relatives. If we find someone deceased on that list, that can be very helpful because then we can go look for the obituary for that individual. If it's a woman and she appears to be married, then we might look for older relatives with a different surname because perhaps she's using her married name and we need to identify her maiden name and then look for who her parents were. And sometimes, as I said, they are associated and you can see they're deceased. That could lead to their obituary or even a published public family tree that can really help us be efficient in building out that tree. So if no parents are associated, then we might search newspapers.com, Genealogy, Bank, and Google for obituaries or other articles. Once we've successfully identified that person's parents, we're going to build their family tree. Once we get back before about 1950, we can start hooking into or connecting to traditional genealogical records like census records. And then we repeat that for the next match in that genetic network and hope they're going to converge at a common ancestral couple. So in April's case, I had a lot to work with. Even though these matches were not close, I had a lot of matches around the second to third cousin once removed level. And so I was able to build the family trees, matches 1 and 3. You can see in the red outline boxes, those are my matches. All these matches shared DNA with each other, 1, 3, 8, 9, and 12. Matches 1 and 3 were siblings and 8 and 12 were siblings. I built their family trees back and found that they both descend from a man named John C. Green up there in blue. He had 2 different marriages. So 1 and 3 descend from his first marriage and 8 and 12 descend from his second marriage. Now, because matches 1 and 3 share significantly more with our subject than matches 8 and 12, it's likely that the subject's going to descend from John C. Green and his first wife, Mary Jones. To further support this genetic network, we had match number 9, and match number 9's tree connected one more generation back. So at John C. Green's parents, Henry Green and Sarah White. So this was my first genetic network with my common ancestor identified as John C. Green, born about 1836. So it's very likely our subject also descends from John C. Green. I had a second grouping or cluster for a genetic network. Matches 2, 4, 13, and 14 shared DNA with each other, but not with my first genetic network. When I built their trees back in time, they all converged at this couple up here in green, James Miller and Nancy Barnett. And so this told me that our subject likely also descended from this couple. So this was my genetic network number 2. So now I have 2 pieces of our unknown suspects family tree. I had these 2 matches, matches 5 and 11. They didn't share DNA with any of those matches in the first 2 genetic networks. I built their trees back because they shared DNA with each other. And I found that they both descended from Henry McIntosh and Rachel Mays up there in magenta. So this is likely another set of ancestors in our unknown suspects family tree. So now we have 3 pieces of his family tree. And finally, I had matches 6 and 7 that shared DNA with each other, but none of my other matches. So they didn't share with genetic networks 1, 2, or 3. And so when I built their tree back, I found that they descend from a couple named Wright Dillion and Avery Cantley. Match number 7 descends through their son, OK, and match number 6 descends through their son, Otay. So we know that the subject likely also descends from this couple. So now I have 4 separate genetic networks and 4 likely ancestors in the subject's family tree. Now, we all have unique family trees except for our full siblings. So if I can figure out the one family, the one person or set of siblings that descend from all of these ancestors, I should be able to zero in on potential persons of interest in this case. So the next step is called descendancy research or reverse genealogy. Now, instead of building backward in time to identify long dead ancestors, I'm now going to flip that tree upside down, build forward in time, and try to identify the descendants of each of these sets of common ancestors. I'm looking for triangulations between their descendants, or typically marriages that lead to births. So it could be out of wedlock. Hopefully it's marriages because those are easier to find because of documentation. But we're looking for at least 2 of these genetic networks, descendants, to marry and have children or just have children. So do the descendants of genetic networks 1, 2, 3, and or 4 converge through a marriage or a birth of a child? So as I built genetic network 1 forward, so John C. Green, I identified all of his descendants from his first marriage and his second marriage, just in case. And I found that from his first marriage, he had a son named Warren Green, who had a son named John Green. So they're in yellow. Who did John marry? So my next step when I'm building these forward and identifying the descendants is I want to find out who did each of his descendants have children with, and then build their trees back to see if they connect to any of the other genetic networks. So John Green married Dora Dillion, they're in yellow. When I built her tree back, I found that she descended from Wright Dillion and Elizabeth Dillion. And her father was Henry Dillion, and he was the brother of Wright Dillion. So this is my genetic network number 4. And so they are converging through this marriage, Dora Dillion and John Green. So any children that this couple has are going to be descended from both my genetic networks 1 and 4. So notably, they had a daughter named Patricia Green. Now I still have 2 more genetic networks that I need to account for. So let's see if those converge. When I built forward from genetic network number 2, James Miller and Nancy Barnett, I found that their son, John Miller, they're in orange, married a woman named Nancy McIntosh. When I built her tree back, it converged with Rachel Mays and Henry McIntosh. So my genetic network number 3 common ancestral couple. So when Nancy and John had children, that brought together the ancestral DNA from my genetic networks number 2 and 3 common ancestors that you can see there in green and magenta. So any children or grandchildren or any descendants of theirs would have the correct ancestral mix for half of my genetic networks. So obviously we need to get all 4 of these genetic networks combined. Well, they had a grandson named Robert Miller that you can see there in yellow. And guess what happened? Robert Miller, the descendant of genetic networks 2 and 3, married Patricia Green, the descendant of genetic networks 1 and 4. So now all 4 genetic networks have converged through this marriage of Patricia Green and Robert Miller. Therefore, anyone descended from them would carry the correct ancestral mix to be related to all of our top 14 matches. That is very, very promising. And look what happened. As is so common in investigative genetic genealogy, location is really key. In about 99% of our cases, I would say, the genetic genealogy starts out all over the country, all over the world sometimes, and it leads us right back to the scene of the crime. And that is what happened here. After this couple married, Robert Miller and Patricia, they moved from Miami to Fort Wayne, Indiana. So now we need to look at who are their sons. Based on their age, their ages and when they were married, it's likely that the subject is going to be one of their sons. I identified 3 sons for them. We'll call them brother number 1, number 2, and number 3. So any of these men would carry the correct ancestral mix to be our DNA contributor or our suspect in this case. So looking more closely at them, I was able to eliminate brother number 1. Why? Well, he was alive at the time of the crime in 1988, but by 2004, when those notes were being left with the biological material included in those plastic baggies that the suspect left around, brother number 1 had died. So it wasn't possible for him to be our suspect, since it was confirmed that that was the same DNA in those notes as from the crime scene. So this took us down to brother number 2 and brother number 3. Now, this is as far as we can go with with investigative genetic genealogy, because these brothers have the exact same family tree, the exact same ancestors. So they are both going to be related to all of those top and key matches that I showed you near the beginning. So the hypothesis that I presented to the agency was that the subject is highly likely to be one of 2 brothers. They are John D. Miller, born in 1959, and his brother. I've privatized his name. And this potential identification was of very high confidence because I had a lot to work with. The match number 1 ended up being a second cousin on their mother's side. Now, remember, that's pretty low sharing. There was only about a 10% likelihood they would be as close as a second cousin, but it turned out that they were. Match number 2 was a third cousin on their father's side. And I was able to tie matches to 3 out of 4 of their grandparents' ancestral lines on multiple unique lines of descent. And the only grandparent line that I wasn't able to tie to the matches was from a family of recent German immigrants, which is typically an origin that results in a decrease in matches in our database. We don't have that many Germans in our databases, therefore we don't get a lot of matches there. So this was a highly confident potential identification. But remember, investigative genetic genealogy is only a lead, as Mark pointed out. And so at this point, my work is done. This is John D. Miller, the amazing Detective Brian Martin and his team then took this lead that I was able to help provide through genetic genealogy, and they investigated these 2 brothers, and they very quickly narrowed it down to John D. Miller, who you can see here, which, by the way, this was the snapshot phenotype prediction from Parabon on the left. Very, very close. The traits are very consistent, but remember, we can't account for somebody's lifestyle choices, including their dental care. So it's not meant to be a photographic likeness, but you can see that the traits are very consistent with Mr. Miller. So In this case, all 4 genetic networks converged through the marriage of Patricia Green and Robert Miller, and John D. Miller was determined to be the DNA contributor based on abandoned DNA that Detective Martin and his team collected and compared against that original STR profile that is used to arrest someone, charge them with a crime, and eventually convict them. The investigative genetic genealogy is, again, just a lead, same as if I were to call in John D. Miller's name through Crime Stoppers. They have to do their full investigation and refute or confirm my hypothesis. So here's a different way of looking at John D. Miller's tree. He's on the left. His parents then are above him, Robert Miller, Patricia Green. And if you follow his tree back, you can see how each of these genetic networks connect to his family tree. We are back at great-great-grandparents on 3 of these genetic networks, and we had to go back to 3rd great-grandparents on genetic network number 3. You can see how these matches and genetic networks connect through his mother's father's side, his mother's mother's side, and his father's father's side. So July 15th, 2018, John D. Miller was arrested for the kidnapping, murder, and rape of April Tinsley. He ended up pleading guilty December 2018, and this was the first investigative genetic genealogy conviction. So although he wasn't the first suspect identified through investigative genetic genealogy, he was the first one convicted. He was sentenced to 80 years. So June 22nd, 2018, genetic genealogy began. July 2nd, 2018, a potential suspect was identified. July 15th, John D. Miller was arrested. December 7th, he was convicted. So the traditional investigation took 30 years, led to hundreds, literally hundreds of persons of interest, many of whom were DNA tested over those years. And the genetic genealogy investigation only took 3 weeks and led to 2 persons of interest. So you can see how efficient this type of investigation can be. Though we're focused a lot on the arrests and the identifications, the real power of investigative genetic genealogy, in my opinion, is to eliminate the vast majority of the population from ever being suspected of a crime, from ever having to be investigated in this type of crime. But you need people with high skill level to be able to make the best use of this data. I had a lot to work with, as I said, but these were distant matches. And so the skill level is important. The higher the skill level of the investigative genetic genealogist, it leads to less persons of interest in an investigation. So it's important to use highly skilled and qualified investigative genetic genealogists. We're dealing with many challenges: recent immigration, diverse population groups, endogamy and pedigree collapse, lack of public vital records in some areas. So experience really matters. We're also dealing with people who have a lot of family mysteries. It's very common that our top match or matches have significant family mysteries of their own that have to be solved before we can use them to help solve the cases we are working on. And so you have to be able to recognize these signs and know how to resolve these. We're also working with degraded DNA, contaminated DNA, and mixed DNA samples. These are not optimal DNA samples. And so we have to understand the impact on the data and how that might change the levels of sharing. Unfortunately, there's still no licensing, certification, or degree for investigative genetic genealogy. It came from the citizen scientist world. And so it's taken a while for everyone to get their arms around this. I'm very excited about the Investigative Genetic Genealogy Accreditation Board that has been formed. I was one of the founding board members, but have stepped away just to because I'm so busy. And this is an amazing group of people that are handling this now. And so they don't need me to be part of it anymore. It's a nonprofit effort, a committee of experts, and they're promoting standards, best practices, and competency. So hopefully they will be able to work closely with the authorities in helping them identify who are qualified genetic genealogists. So what does the future hold for investigative genetic genealogy? Well, when we can use it earlier in an investigation, certainly we can have the most impact. And we're seeing that happening across the country now as law enforcement has really grasped the power of this tool. We can stop criminals in their tracks. We can perhaps eliminate serial killers and rapists because we can identify them after their first violent crime. Hopefully this will save lives. I believe it already has, even if we'll never know exactly who and how. And maybe even it might deter some would-be criminals, knowing that it is virtually impossible not to leave their DNA behind with these types of very violent, intimate crimes. And if they leave their DNA behind, they will be identified through investigative genetic genealogy, whether it takes a couple days, weeks, or months, sometimes years, it will happen. We're seeing very quick technological advancements now that genetic genealogy is being used for law enforcement. Cases that were deemed unsolvable in 2018 or 19 or even 2020 are now workable and perhaps solvable. So this is continuing to advance at a rapid pace. Now, with this immense power of genetic genealogy comes a deep responsibility. This tool relies on public support and public trust. And that means that we need to not only be performing it with competency, but also with high standards and high ethics. We need to develop and follow best practices. We also need to educate the public and dispel the many misconceptions about IGG so we can ensure that they're making informed decisions, whether they decide to actively support what we are doing by providing their DNA in the databases that we are able to access, or if they decide not to. We want them to do that from an educated standpoint and not from fear-mongering and misconceptions. So the benefits of genetic genealogy for law enforcement are we eliminate the need to interview hundreds or even thousands of persons of interest in these cases. We can conserve and focus agency resources for more efficient investigations, make society a safer place, provide answers for the families of victims and surviving victims of violent crime, and those who have missing family members and loved ones. I don't know if we have time for questions, but thank you so much for your attention. It's been my honor to speak today, and I hope that you learned something from it.
Thank you, CeCe, and thank you, Tony and Mark. So we're going to push back break a little bit because I know there's a lot of information here. Any questions? Thanks for waiting.
Hi, and I'm not sure who the best person to answer this question from the panel is, but I'm in Washington State and I work with our SAKI program, and we've done a lot of funding for forensic genetic genealogy. And as you know, we had the first trial in our state related to forensic genetic genealogy. So we have some court challenges to it. And the most recent one, I listened to, there's a case in the Court of Appeals, and I listened to the argument, and the defense attorney, I mean, it didn't, in my opinion, it didn't really seem to have a lot of legal basis, but she was kind of capitalizing on this, you know, like you said, I think fear mongering, this sort of fear of this dystopian future where the government has access to every little part of our being. So my question is, and she mentioned, I guess, a database called, is it Prometheus or something like that, where people can take their profiles and upload them, and then that will tell them what their percentage of having a particular disease is or whatever. So my question is, how do you assuage the public's fear that with the information that law enforcement gets from this sort of stuff, that then they wouldn't have the ability to find out like what diseases people might have or you know, other things about their being that the public doesn't necessarily is afraid, I guess, of them knowing?
I think I can address this, at least to start. Now, that is why I do a lot of media and interviews. I actually don't enjoy it, which might surprise people, but I'm doing it because I'm working very, very hard to dispel the misconceptions out there and educate the public about what investigative genetic genealogy is and what it is not, what we do and what we don't do. And I didn't have a lot of time to talk about it today. I typically do talk about this in more detail, including the first jury trial that you alluded to. We're not looking at the genetic code, as Mark said. We're not looking at the ATCs and Gs. We only have access to how much DNA is shared between two people. And if we really want to, we can see what chromosome and what location that DNA is shared. So we're not looking at their medical records, their risks. And so, yes, it is possible to upload a file to Prometheus or many other places, but that's not something we're doing in investigative genetic genealogy. We're uploading to these two databases that just provide these match lists. So it is a major undertaking to try to dispel all of this fear mongering. And I think it comes down to education. The more we can educate the public, the more we all can get out there and try to dispel these misconceptions and the fear mongering, the better. Sometimes I feel like a one-man band out there, and I need an army of people that are spreading the word about this and helping to educate. As far as in the courtroom, Parabon, my team has been involved in over 60 convictions already. And I think most people don't realize that. They think there's been no case law or very little case law or court precedent, but we've already been able to help set a lot of precedent about the use of investigative genetic genealogy. So any district attorneys, prosecutors that are involved in a case like this should really reach out to a district attorney or prosecutor who has been involved in one of these cases. And so in your case, there's an amazing DA who worked on the Talbot case, Jay and Tanya, and Jay and Tanya's
Matt Baldock, is that who you're talking about?
Yes, exactly. So
he's a friend of mine.
Oh, well, great. I think he's one to really address those questions. We also have a legal liaison at Parabon who has been helping prosecutors and detectives even how to write up these warrants to make sure we're not using IgG as a basis for an arrest or a DNA collection. So you can always reach out to us and get some advice on how to present IgG in this context.
So I know you guys don't, upload it to Prometheus, and I know that you're not looking at people's medical history or potential, risk for some sort of genetic disorder. But my question is, I guess you could, right? You do have the ability to do that. And I think the fear kind of goes more towards law enforcement doing that. So does law enforcement get enough data to be able to do that? I mean, I know the private labs have enough data to be able to do that if they wanted to.
I mean, I think it's different with different providers, but typically with Parabon, they're not getting the raw data file, which is what you would need to upload. They can certainly request it because it does belong to them. That has already been, debated and worked out and decided. So yes, we could present it. So this is why I think this is a much bigger topic, but this is why I think legislation is so important. And I've been closely involved with some legislation, particularly in Utah, to set these best practices and codify into law the things we should be doing and should not be doing, and make them actually illegal to do. And so in Utah, you would not be allowed to upload that file to Prometheus. I think we need a federal law. The DOJ guidelines are great, but we need it codified into law to make sure that everyone is following the rules and the best practices.
We have time for maybe one more question.
Excuse me, I have a question for Mark. Mark, you made a statement, and I apologize if this isn't a direct quote, but you said that the FBI interim policy needs to be abided by, or should be abided by, for the database policies, or we should be following the database policies, essentially. I'd like some clarification, or if you want to choose between should be or must be. And then also, what would you say to those that feel that the DOJ interim policy is merely a suggestion and not policy?
Well, I think the policy, the initial policy was just a set of guidelines. So it has no legal backdrop to it. So yes, they should be abided by. In terms of the until they're codified into law, which is yet to be done. But in terms of the terms of service, these are private companies that have rules about how their databases are to be used. Now, these private companies expect that law enforcement agencies and anybody else will abide by these terms of service. A lot of companies do, but there's some potential information out there that from time to time, those terms of service are not abided by. There's uploads that are done outside of the terms of service. But since they're a private company, there's no legal recourse to do anything about that, right? So in the future, that's what we need to think about. Are these databases used in the right way? And does the public have confidence that we are doing it correctly? And one of the things that they're going to ask about, the public, is, are the terms of service being abided by? Or is law enforcement and others actually abiding by the stated terms of service of these companies? They're private, but there's no legal recourse to back them up. Does that address your question?